chromatin function is one of the most active areas of biomedical research. It plays major roles in controlling gene expression and is regulated by a complex interplay of chromatin-associated proteins and histone post-translational modifications, or PTMs. Defects in chromatin regulation are observed in nearly every type of human disease, including cancer. Mapping of local chromatin features is thus essential to understanding chromatin regulation and its role in disease. Although much progress has been made, further advancements are limited by the power of current technology, namely chromatin immunoprecipitation, or ChIP. But a new approach could make the problems with ChIP a thing of the past. CUT&RUN, short for cleavage under targets and release using nuclease, builds on the chromatin immunocleavage, or ChIP approach, to provide researchers higher resolution with lower cell input and sequencing requirements than previously possible. To appreciate this new tier of performance, let's review how ChIP works. In ChIP, large pools of fragmented chromatin are treated with an antibody that binds to a chromatin-associated protein, or histone PTM. The antibody is coupled to magnetic beads, which enables isolation of antibody-bound chromatin. Associated DNA is then purified and prepared for targeted or genome-wide analysis by next-generation sequencing. Despite their popularity, ChIP assays are time-consuming and provide fairly low-resolution data with high background. Increased cell inputs and deeper sequencing analysis can partly overcome these deficits, but add up to an expensive price tag and do not guarantee consistent or reliable results. That's where CUT&RUN comes in. In CUT&RUN, cells are immobilized onto magnetic beads and permeabilized. Cells are then treated with a factor-specific antibody, which then further recruits a protein AG and micrococcal nuclease fusion. This PAG-MNase, tethered to specific genomic binding sites, is activated by the addition of calcium to cleave DNA. The resulting chromatin fragments diffuse out of the cells, and the protein-bound target DNA is extracted and sequenced. Everything else remains inside the cell nucleus and is removed with the magnetic beads. By selectively releasing antibody-bound chromatin and only sequencing these regions, CUT&RUN dramatically improves the low signal-to-noise ratio and high variability that plagues ChIP assays. This allows for much lower cellular input, 5 to 10-fold reductions in sequencing costs per sample, and shorter turnaround times. As a result, CUT&RUN has experienced rapid uptake by the chromatin field, and the number of papers citing this technology are steadily increasing. With the release of the Katana platform and products, EpiCypher is now providing this capability to all scientists, poising CUT&RUN to have a major impact on the field.